Alright, so here we are on the second of the three kinds of filaments, or fibers, that we find in the cytoskeleton. This one we're going to focus on what are called the intermediate filaments. And there's a couple of things that are unique about these. And probably the first one is that these are solid as opposed to the microtubules, remember, that were hollow. And these are fairly flexible, so they bend fairly easily and would be fairly easy to, I mean, you can't crush them, but they just don't offer a whole lot of resistance. But these intermediate filaments are very strong when it comes to pulling them apart. And so, like I said, they're kind of the opposite of the microtubules. The microtubules are these hollow tubes that are keeping you from crushing the cell. The intermediate filaments are going to prevent you from pulling it apart. So it's almost like these cables that are running back and forth throughout the cell. And you'll see how it kind of connects cells together as well as we move into other material. The other thing that is somewhat unique about the intermediate filaments is that they tend to be variable as to what they're made out of. So whereas microtubules are made out of tubulin, intermediate filaments, depending on the kind of cell you are going to make your intermediate filaments out of specific kinds of proteins. And each one of these proteins will have slightly different properties. Some will be tougher and more rigid, some will be a little bit more flexible, a little bit more elastic, so that, again, you're trying to protect against pulling forces, and you're going to want some resistance to that. And some tissues are going to resist a lot, some tissues will resist a little bit less. Last known, there were at least 50 types of these intermediate filament proteins out there. Some that you guys have probably heard of before would be keratin for sure. Vernon would be another one. Keratin you're probably familiar with in the fingernails and the hair, and Vernon would be found in other places in the body in different ways. The function, like I said, is largely structural. Not much else going on here, although it's not so much structural in making sure the cell keeps its shape, because that's the microtubule's job. In this case, it's more about the tissue and making sure that cells that are supposed to stick together and stay together actually do stick together and stay together. So that you're not going to, like if we've got a liver, then we're not going to be able to just pull that liver apart, or that liver's not going to fall apart, because the intermediate filaments inside those cells are going to hold us together. And the way this works, and again, we'll get into more details on this later as we think about what ties cells together, is if we had some cells, then the intermediate filaments, so rather than radiate out from the center, these guys go all the way across. Like I said, they're almost like these just cables that run from side to side throughout the cells. And what we'll see is that there's going to be contact points where these cells link together, and you'd have contact points all around with all the other cells in the tissue. And these guys are going to basically tie us all together. So how does this increase the structure and the safety of your tissue? Is it would be impossible to basically pull these apart, okay? Because if you pull here, these cables, not only do they keep you from pulling this cell and damaging it, they keep you from pulling it away from these cells as well. And then of course these guys would go off into other cells, and each cell is responsible for its own fibers. So this is not one continuous. These are actual building blocks or contact points that we'll talk about when we start putting cells in in the context with each other. But as we pull in one direction, what it's going to do is it's going to basically submit that force through all these cells, and the tissue will either give a little bit, but it just makes sure that you don't tear apart the cells and you don't tear apart the actual tissue as well. And of course, not to give a whole lot away yet, but the extracellular matrix is all in here as well, and is going to make sure that this happens as well. Okay, so that's a part of the story we'll get to later, is the extracellular matrix also tying into this process. Okay, so depending on the tissue type, you may have different properties to these cables that are running through the cell, and let's just take a look at them. So again, somewhat different from what we saw before with the microtubules, so not really radiating out from the nucleus to prevent us from being crushed and protect that nucleus from being crushed, but these cables basically run across, and notice how they go to the surface, go to the surface, go to the surface, go to the surface, and converge on these little spots where we would be connected to the cells next door. Okay, so things like fingernails and hair would use the keratin in a different way. These would be cells that are using keratin as the intermediate fiber, and really there to prevent us from being pulled apart. Okay, so next time we'll talk about the microtubules, the microfilaments, and finish off the cytoskeleton material.